What's going on, everyone? Today we're going to finish up our conversation on how neurons communicate with one another. We're going to talk about action potentials, and then we're going to talk about neural networks and neural circuits. But before we get into all that, let's just have a little review from the last lecture. One, our nervous system is divided up into multiple kinds of cells. We have the neurons, which are actually going to send chemical messages throughout the body. And then we have neuroglia, which help assist the nervous system in maintaining the blood-brain barrier and with phagocytosis and just cleaning up debris and things like that. But these neurons are going to be the ones that communicate with one another through electrochemical signaling, the movement of ions and charges, in order for our bodies to function. And so in order to do that, we need to have ions move through what are called channels, because once those ions move, they are going to bring a current with that movement of ions if there is enough movement. And so when this happens, there is a change in membrane potential, which is the charge of the cell's membrane during certain periods. So let's talk about neurons and their communication. So if we wanted this neuron in the blue to send a message and release neurotransmitters here at the synapse, well, it needs to get to that point of threshold like we discussed last time. And so how is that going to happen? Well, it's going to happen through graded potentials. And so the neuron that precedes it, if it sends out an action potential and neurotransmitters are released, they're going to bind to receptors on the dendrite of this neuron. And now these neurotransmitters, they could either be depolarizing, which means we are getting less polar, so this negative membrane charge of this neuron right here is going to become more positive. But neurotransmitters could also have the opposite effect, and they could hyperpolarize, meaning they make the membrane more polar. And so if we want this membrane, if we want this neuron to actually fire its neurotransmitters, we need to reach threshold, and in order to do that, we need to depolarize. So certain neurotransmitters will bind to those receptors. Ions will enter the neuron and depolarize it. And then again, if we reach that threshold, that negative 55, that message is going to be communicated down the axon, and then neurotransmitters are released. But let's dive into a little bit deeper about how that transpires. So the action potential has three phases: a depolarizing phase, a repolarizing phase, and then an after hyperpolarizing phase. The depolarizing phase is going to be the rising phase of the action potential, where the membrane potential is going to go from negative 70 millivolts, and then again the stimulus has to be strong enough in order for it to reach threshold. So enough ions need to enter the cell here in order to reach that negative 55, and then again it's all or nothing. Once we reach that level in the membrane potential, this depolarization is going to occur. And so what is going on during this depolarization phase? How come we're going from negative 55 on our membrane potential all the way up to positive 30 millivolts? And the reason for that is because voltage-gated sodium channels are going to open, which are going to allow sodium to come from outside of the cell, from the extracellular space, into the cell. Here we see, this sodium channel. And once we reach that threshold of negative 55, this voltage-gated sodium channel is going to open. And so those sodium ions are going to flood into the cell along with their positive charges. And what happens then, that's going to make the membrane potential increase. So we're depolarizing here. And so once we depolarize and get to that positive 30, these sodium voltage-gated sodium channels are then going to close. And then voltage-gated potassium channels are going to open, and that's going to start our repolarization phase. And so when potassium channels open, what we see is that the potassium is going to go from a high concentration from the intracellular space to a lower concentration in the extracellular space. So these ions are also positively charged. So when positively charged ions leave the cell membrane potential is going to become more negative. So that's what we see. The charge will go from that positive 30 all the way back down to that negative 70 level. And then we're going to get a final little phase called the after hyperpolarization phase. In the after hyperpolarization phase, what we're going to have is that these voltage-gated potassium channels are going to remain open a little bit past the resting membrane potential. And then the membrane potential is going to reach that equilibrium potential for potassium. And so those channels are not going to close until around negative 90. And then once they do close, then we will return back to the resting membrane potential. What we're going to see is that once we reach threshold, that action potential is triggered at a place in the neuron called the axon hillock. And so once that occurs, then the depolarization is going to continue all the way down the axon until we reach that synaptic end bulb, and then neurotransmitters are released. So here you sort of see how that action potential is traveled along that axon. So we have those voltage-gated sodium channels opening. We're going to have depolarization of this section of the axon, and then the neighboring sodium channels next to it, once that depolarization occurs, they're going to open. And so that depolarization goes from one section to the next because those voltage-gated sodium channels are opening further down the neuron. And then once the section before is depolarized, it reaches that positive 30 millivolts. Well, then that's when those voltage-gated potassium channels are going to open. Potassium leaves the cell, repolarizing, and then you have that slight phase of after hyperpolarization before returning back down to resting. And so this depolarization is going to go throughout the axon, followed by the repolarization. Once an action potential reaches the synaptic end bulb, that depolarization is going to cause voltage-gated calcium channels to open, and then calcium is going to enter that place in the cell. And you don't really need to know this level of detail, but basically calcium is going to activate a protein called CAMK2, which is going to allow these vesicles to start to move towards the presynaptic membrane, and then these vesicles are going to fuse, releasing the neurotransmitters into that synaptic cleft. Those neurotransmitters can then bind to receptors on the postsynaptic neuron. And if those neurotransmitters lead to a graded potential that then causes the next neuron to reach threshold, well, then that next neuron is going to fire its own action potential. In order for neurotransmitters to be released, like I said, these synaptic vesicles have to bind to the presynaptic membrane of the presynaptic neuron. And so what's going to happen is you have these proteins called snare proteins. And with the help of calcium as a second messenger, it's going to help activate synaptotagmin, so where this vesicle can then come in close proximity with the membrane, and then these snare proteins can then fuse together, allowing for this vesicle membrane to join with the cell membrane. And then when that happens, this gap opens, which then releases those neurotransmitters into that synaptic cleft. Like I alluded to earlier, not all neurotransmitters are going to lead to an action potential in the next neuron. Some neurons actually serve to inhibit the communication of the neurons following it. And so we have what's called inhibitory postsynaptic potentials, where neurotransmitters cause hyperpolarization, right, to try to inhibit the action potential of the next neuron. And then we have neurotransmitters that cause excitatory postsynaptic potentials, where those neurotransmitters will lead to a depolarization on the membrane of the postsynaptic neuron. A lot of neurons and cells in our body are receiving both inhibitory messages and excitatory messages. And basically, whether or not an action is going to occur is going to be based on the amount of inhibitory neurotransmitters and the amount of excitatory neurotransmitters. And so for this neuron, in order to fire an action potential, those excitatory potentials have to outweigh the amount of inhibitory signaling. And so again, if we get enough excitatory signaling and enough ions can depolarize and we reach that threshold, an action potential will be triggered. Neurotransmitters can bind to two main receptor types. We have one that we've been talking about a little bit more in depth with, and that's ionotropic receptors. And from the prefix here, we could see that there's going to be a movement of ions through these receptors. So when that neurotransmitter is released and then binds to these ionotropic receptors, they're going to open, allowing for ions to move. And then along with those ions, we're going to get graded potentials. Here you can see one example of a neurotransmitter and its receptor. So this neurotransmitter, acetylcholine, is released and has many different functions depending on what cell it is going to bind to. But this ionotropic receptor, we call those nicotinic acetylcholine receptors. But acetylcholine could also bind to different types of receptors. And so here we see what's called a muscarinic acetylcholine receptor, which is a type of metabotropic receptor. And metabotropic receptors function through a G protein, which we learned about in previous lectures. And so when acetylcholine binds to one of these receptors, that G protein is going to get activated, and then we get the same result in the end. This G protein will then have an effect on maybe, let's say, another ion channel. So in this example, the muscarinic receptor is activated, that G protein is activated, and then results in this potassium channel opening, which causes an inhibitory postsynaptic potential. So again, just keeping in mind that these neurotransmitters, they can bind to ionotropic receptors, where ions are going to move through those receptors, or metabotropic receptors, where a G protein will be activated and then lead to a signaling cascade, which eventually leads to ions moving in or out of the cell. So if we want a signal to stop from one neuron communicating with the next cell, you need to get rid of the neurotransmitters. And there's a couple of ways to do that. So either these neurotransmitters can be diffused out of this membrane space and then just enter the extracellular space, where it could be broken down by things like astrocytes. We also have enzymes that can break apart neurotransmitters down to smaller particles. And then we also have reuptake or recycling of the neurotransmitter. But either way, no matter how you get rid of the neurotransmitter, once you get less concentration in that synaptic cleft, less is going to bind to the postsynaptic membrane of the next neuron, less will bind to receptors, thus terminating the signal. Remember that neurons are a vast network, and even one neuron can synapse with multiple different neurons at many different places. And so there could be regulations that occur in the presynaptic membrane, that synaptic end bulb, in order to modulate neurotransmitter release. So here we see three neurons, neuron A, neuron B, and neuron C. And neuron A is trying to communicate with neuron C by secreting a neurotransmitter, right? But neuron B can impact right here because it's making an axon to axon synapse with neuron A. It could ultimately affect how much neurotransmitter is released from neuron A to neuron C. And so let's say that neuron B has an action potential and then releases its neurotransmitters, which bind to receptors on neuron A. It could inhibit the release of the neurotransmitter secreted from neuron A. But also, neurons could emit excitatory potentials, which then bind to receptors on this synaptic end bulb of neuron A, which then enhance the release of neurotransmitter. So basically, there is regulation in all fronts in order to either amplify a signal or to try to lower the communication that a neuron emits. There are multiple kinds of neurotransmitters, and each have different effects. Even one neurotransmitter can have a different effect depending on what type of receptor it's binding to or type of cell that it's binding to. But the response from that neurotransmitter is going to be amplified when more is released, because when more neurotransmitter is released, more will bind to receptors on the postsynaptic membrane. I do want to just call out a couple of these because they're going to be important ones that we discuss in the next few chapters, talking about the nervous system. Two are going to be epinephrine and norepinephrine, or commonly known as adrenaline and noradrenaline. And so these are going to be the neurotransmitters of the sympathetic nervous system, which you may know as the fight or flight side of our nervous system. So when we go on a run, when we start physical activity, that fight or flight response turns on, and these neurotransmitters are released and bind to receptors, which pick up our heart rate, which pick up our blood pressure, and get us ready for that physical activity. We also have a neurotransmitter called acetylcholine, which is the neurotransmitter of the parasympathetic nervous system. So that's going to be the one that slows our heart rate down. But acetylcholine, that's not its only function. It's actually going to play a role in the contraction of our skeletal muscles. It's going to be released and start the cascade, which starts a skeletal muscle fiber contraction. But I will get more into those in future lectures, but I just kind of want to point them out now, just to keep in the back of your head that these are going to be very, very important neurotransmitters going forward in future lectures. We're going to conclude with the different types of neural circuits. There are anatomical setups for neurons in order to communicate messages throughout the body. And so the first one we'll talk about is called a diverging circuit, where one neuron, where a single presynaptic neuron will innervate multiple postsynaptic neurons. And you're going to find these frequently in both sensory and motor systems. Another type of circuit is going to be called a converging circuit, which is essentially the reverse of the last one. So now we're going to have multiple neurons that synapse with a single neuron, and some of these neurons may be sending inhibitory messages. Some of these messages may be sending excitatory messages. And again, all of these signals are going to be weighed against each other, and whichever one is winning out at a particular moment will dictate whether or not an action potential is sent in this neuron. A reverberating circuit is a circuit that's commonly used in repetitive motions, such as locomotion, so walking or running. So what you're having is neurons are going to fire and signal down this circuit, but then there are basically checks in communication where neurons later on down the circuit can regulate firing in previous neurons. And so there is regulation going in the forward direction, and then there's regulation from these neurons going in the reverse direction. And then we have what's called a parallel after discharge circuit, where a single neuron is going to basically activate multiple groups, parallel groups of neurons, all which have a single common postsynaptic neuron. That is going to conclude today's lesson. I want you guys to make sure you go back and memorize the steps of the action potential from reaching threshold to the depolarization, repolarization, and after hyperpolarization phase. Just know which ion channels are opening during those periods, and then also making sure that you understand how neurotransmitters are released and the different types of neural circuits. Thank you guys so much, and we will be continuing our discussion on the nervous system, and we're going to talk a little bit more about the different branches, including the autonomic nervous system. We'll talk a little bit more about the brain and the sections of the brain and how it handles certain functions of the human body, and then we'll end off the nervous system by talking about motor function and the somatic nervous system. So thank you guys so much, and I'll see you next video.